So this class is about bacterial genetics, and I hope many students are aware of um, the discovery of DNA was by Watson and Crick, and you guys must have studied right from the school time. Then right from the first year, you would have known about the structure of the DNA and then RNA, everything. So I'll rush through all those things, and then we will concentrate about bacterial genetics. So okay, so these things, all these words also you must be familiar, like in the molecular biology, like transcription. Transcription is when DNA is transcribed to RNA, and translation is when RNA is converted to polypeptides. So these words also you must be familiar, and how RNA differs from DNA also you all must be very much in knowledgeable. So here only the sugar uracil is replaces the thymine, and then there are three types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. So now we are coming to the bacterial chromosome. It is actually again a double-stranded structure, DNA, and it is a circular form. And the length is about 1000 microns, and it contains around 4000 kilobases. So it is like human have about 3000 base pairs, but then bacterial chromosome have 1000 base pairs. So how bacterial genome differs from higher forms of life is mainly several stretches of DNA don't appear to function as codons, and those parts are called as introns. And then the coding part of the bacterial genome is called as the exons. So in transcription, these introns are excised when form RNA before translated by the ribosomal protein. So these terms also, I hope you are all very familiar. Exons is stretches of coding genes, and then introns, these are coding sequences of genes that are useless and non-functional. So during transcription, both are copied, but only during translation, exons of the RNA are translated, and then introns are excised. And there is one important different feature from that human genome. The bacterial genome possesses some extra chromosomal genetic elements. So I hope you are all familiar with the extra chromosomal genetic elements. And then these are actually not essential for actual survival of bacteria, but then it helps the bacteria to become resistant to antibiotics and makes them survive, and also they helps them to produce virulence structure called as the, or virulence agent called as the toxins. So these extra chromosomal genetic elements are otherwise called as plasmids. So as I have told, they are circular DNA molecule, and they are present in the cytoplasm, as you can see, and they are capable of autonomous replication compared to the DNA molecule. And then they can transfer gene from one cell to another as well. They act as a vectors on genetic engineering, and they are also present in yeast. Yeast is a fungi. So these plasmids are not only present in bacteria, they are also present in fungi called as yeast. So you can see here the bacterial structure, and this red structure is the bacterial DNA, and the blue color are plasmids. There can be more than one plasmids in a bacterial cytoplasm. So this is the question. All are true of plasmids except. So choice A is smaller in size, they are extra chromosomal, they are circular, and they are ribonucleic acid. So here, as you all know, the best choice or the correct choice is ribonucleic acid. All the other choices are correct. So it is an extra chromosomal DNA. Okay, it's not a ribonucleic acid. And then plasmids are not essential for normal functioning of cell. They confer property like drug resistance, as I have told before. But then when this becomes integrated to the bacterial chromosome, it is called as an episome. Plasmid becomes episome. And then they are important vector in genetic engineering. So they are it's presently in bacteria. They encode genetic information for many properties like resistance to antibiotics, bacteriosin production, enterotoxin production, enhanced pathogenicity, and reduced sensitivity to mutagens, and they degrade complex organic molecules. So that much of genetic information are carried by plasmids. And this is structure of R plasmid, or otherwise called as the resistance plasmid. And here it can carry resistance to many drugs at a time. One plasmid can, so as you can see in the structure, it is resistant, it carries resistant genes to carbonicillin, tetracycline, chloramphenicol at a time. So these R genes are called as drug resistant genes, and RTF are resistant transfer factor, or otherwise R plasmids. Okay, this is the structure of the R plasmids. So episomes, as I've told before, they can be integrated with chromosomal DNA, or otherwise integrated form of plasmid with DNA is called as a episome. Okay, so these are the plasmids. So during cell replication, they independently replicate, but plasmid, when they become integrated with chromosome, they are called as episomes. So they are dependent on replication with the chromosome. Simultaneously, they replicate with the chromosome. Okay, so these are the potentials of plasmids, like they are self-transmissible and they can be not transmissible as well. So these are different types of, like when their ability to produce conjugation tube. So conjugative plasmid and non-conjugative plasmid. This is one type of plasmid. So plasmids can be classified as, based on the potential, transmissible or non-transmissible, and they can be compatible or incompatible plasmid, and they are classified based on function. So when in this part of the classification, when they are transmissible, it is called as the transmissible plasmids. Like when they have got, or conjugative plasmid, when they have got the potential to produce pilus or conjugative tube. So in this case, they transfer the sex factor and drug resistance with the help of restrictions on nucleases. And as I have told before, compatibility typing, they can be compatible or incompatible. So which means like it doesn't accommodate other with which are similar. So it fits it only certain type of bacteria and doesn't get into another type of bacteria. They're compatible with certain type of bacterial chromosome, and they are not compatible with certain types. So it is called as compatible plasmid and incompatible plasmid. And this is the functional classification. So it is fertility plasmid, which has got the potential to produce F plasmids, and then col plasmids are the one which produce the toxin, which has got the ability to produce toxins called as colicins, and resistance plasmids, which carry the genes for the resistance for the antibiotic, then degradative plasmid, then virulence plasmid. Okay, so next is so these are the things which you should remember. These are the terms we have to be familiar. What is an exon? What is an intron? What is a plasmid? What is a episome? What are the different types of plasmids? And then what are the uses of plasmids? So these are the gist. You have to be gist of the plasmids, which you have to be familiar with. And then we go to genotypic and phenotypic variations, which again you must have studied in biochemistry and then genetics everywhere. So genotypic variation is which shows variation genetically. Phenotype variation is the physical expression of the environment, which you can see. Phenotypic variation, which you can see. The same as like human beings, higher organisms, genotypic and phenotypic variations are exhibited by bacteria as well. So exhibit phenotypic variation, different phenotypes. Say for example, typhoid bacilli, they can be a flagellated one or non-flagellated one, and they can be lactose positive and lactose negative. So beta E. coli is E. coli is dependent on beta galactosidase. When they got the gene to produce beta galactosidase, then it becomes lactose positive. When it is absent, it turns out to be negative. So these are the different phenotypic expression. So you can see colonially different types of E. coli can be present. And then principles of genotypic variation. Genotypic variations is the variations which are shown uh, genetically. So for which you have to remember what is a mutation, and then these variations can happen due to mutation, and genotypic can be transferred by transfer of genes. So by three mechanism: transformation, transduction, and conjugations. Okay. So first we see mutation. Mutation is a random, undirected, heritable variation. Again, mutations, the different types of mutation, you all must be familiar. So it is caused by, it can be caused by alteration in the nucleotide sequence at some point of DNA, which can lead to addition and deletion. And then it can occur in any sequence, and it is inevitable, and it is useful for survival of the bacteria. So different types of mutation, it can be spontaneous or it can be induced. Spontaneous mutation, and it can be addition, deletion, substitution. Substitution can be a point mutation, and you should be more familiar with what is frame shift mutation, what is missense mutation, what is nonsense mutation. So I'm just giving you a highlight of all the different types of mutation. So which I can't go into each type because I have got only just an hour to complete all the bacterial genetics. And then what is lethal mutation? Then what is a conditional lethal mutant? So it is temperature sensitive mutant, like it is permissive to temperature at 37 degrees C and non-permissive at temperature of higher temperature, that is 39 degrees C. And but then multiple mutation can cause extensive damage. So this is the example of point mutation. The point mutation can be silent, or at one point it can happen, it can be a silent mutation, which means nothing can be changed. And then as you can see, when the thymine is replaced with adenine, still it is able to produce only lysine. So it is all the same. And then nonsense mutation, one of the change, if it is replaced with uracil, it stops. So it becomes a stop codon. So that is a nonsense mutation. And missense mutations. Then frame shift mutations is that alters the reading frame of in which the codons are translated. So it can happen by insertion, deletions, and translocations. And then these are the different mutagenic agents. So UV rays, alkylating agents, and then acridine dyes. So most of the chemicals, okay, these are all mutagenic agents. And how will you see select? You can detect a mutation by a technique called as replica plating, where a velvet covered disc is used to transfer all colonies from the master plate, and incubation is different. So you can identify a mutant. This is a typical way to identify a mutation in the colonial mutation, and this is called as Amy's technique. So next we go to how genes are transferred. So transformation of genetic material is, transfer of genetic material is by different methods. So here is the question. So transfer of genetic information through the agency of free DNA. So this is a method by which free DNA is transferred from one bacteria to another bacteria is called as. So all must be familiar. It is a very easy choice. So this is transformation. Transformation is the way by which a free DNA is picked up by another bacteria. That is how DNA is, bacterial DNA is transferred. Okay. So direct uptake of DNA, and it is first discovered in pneumococcus by Griffith, and then it is all this transformation is seen in bacteria like Bacillus species, Neisseria, Haemophilus, pneumococcus. Okay. So as you can see here, free DNA fragments are lying down after the death of the bacteria, and then recipient take a chromosomal DNA from the environment, and genetic recombination can occur that can produce a genetically transformed cell. So this is called as transformation. Next, a property of acquiring antibiotic resistance from a viral colony as such. So here it is called as transference, a conjugation, or a transduction, or a mutation. So here the key word is viral colony. So your choice should be transformation or transference is by naked DNA. Conjugation is formation of conjugation tube. Transduction is transfer of genetic elements by bacteriophages. Bacteriophages are nothing but viruses that infect bacteria. So here the choice is transduction. Okay. So bacteriophages is viruses that parasitize bacteria. Example, lambdaphage is a bacteria which can affect the E. coli, and it has got two life cycle, lytic cycle, and then temporal lysogenic cycle. Okay. So the three bacterial methods of gene transfer is conjugation, transduction, and transformation. Conjugation, direct transfer from one bacterial cell to another by a conjugation tube. And here transduction is through bacteriophages. Transformation is naked DNA is taken up from the environment by a bacterial cell. So first we will see transformation. As I have told you, that the transfer of genetic element through activity of DNA, like naked DNA, which is present in the environment, which has been released by the bacterial cell. So this is the Griffith experiment. A mice is injected with live non-capsulated, that is a rough pneumococci with healed killed capsulated smooth pneumococci. You can see the Griffith phenomenon. So rough strain is actually a non-virulent strain. So mouse lives, and smooth strain is a virulent strain. So here capsule is a virulence factor of a pneumococci. So naturally the mouse dies. Suppose you heat kill the smooth strain. So the virulent strain will become avirulent, that is capsule is being removed. So naturally the mouse will live. Now you combine a rough strain and a heat kill smooth strain. So what happens here? Rough strain is again separately non-virulent, and heat kill smooth strain is again non-virulent. But when they are combined together and given, what happens is again the mouse dies. How does this happens? Because the heat kill smooth strain picks up the non-virulent strain, the DNA, which has got the capacity to produce, or the genetic material which has got the capacity to produce capsule. So that is picked up, genetic recombination happens, and now it becomes virulent, and that kills the mouse. So this is how the transformation phenomenon is explained by Griffith. Okay. This is again another pictorial demonstration. You can see it later. So next we go to transduction. As I have told you, it is transfer of genetic element, the bacterial DNA by bacteriophages, which are nothing but viruses which infects bacteria. So this is the structure of bacteriophage, which will again be taught in virology. So these are the phage particle. It looks like a space vehicle. Okay. So it goes, this bacteriophage goes and sits with the tail fiber on a bacteria and injects the genetic element. So two types of transduction, lytic and lysogenic, and then that is the lytic cycle or otherwise virulent cycle. So after a large number of progeny are built up, the bacterial are released, host bacterium ruptures and releases the virus, progeny viruses. So that is lytic. And lysogeny, it goes and integrates with the bacterial DNA, and then it is multiplied along with the bacterial DNA. So this is lysogenic conversion, typically seen in diphtheria bacilli, which acquires its toxigenicity by lysogenization with phage beta. So beta phage is a bacteriophage which has got the genetic component to produce a toxin. So when that goes and infects diphtheria bacilli, so the genetic element will go and integrate with the Diphtheria bacilli, and now it picks up the toxigenesis. So the elimination of charge of toxic strain renders it non-toxigenic. These are again pictorial demonstration of lytic cycle and then non-lytic cycle, which I have explained. So lytic cycle, the progeny virus are released by rupture of the bacteria, and then lysogenic cycle, it goes and integrates under how it is released, it separates only under stressful condition. The prophage DNA is separated and then further it is released by lytic cycle. So, and again, transduction can be generalized or restricted. Generalized transduction, it involves any segment of DNA, and restricted when a specific bacteriophage transduces only a particular genetic trait. Okay. So, again, there is next question. An F factor which integrates with the bacterial chromosome results in the formation of HFR, RTF plus R, F prime and RTF. So this will be, I will explain further in conjugation. So as you can see, so here the correct choice is HFR. So when an F factor integrate with the bacterial chromosome, it results in formation of HFR. So plasmids are responsible for formation of conjugation tube are called as F factor, and the cells, bacterial cells which carries F factor, it is otherwise called as F plus cells. When it mates with F minus cells, which doesn't have a conjugation factor, it renders them F plus. So when F factor is in integrated state with the bacterial chromosome, it becomes HFR cells or otherwise high frequency cells. So this happens in a small proportion of F plus cells, and conversion of F plus cells to HFR is actually reversible. I will explain that in conjugation. Okay, so this is conjugation. So it was discovered by Leidenberg and Tatum. And then here it is a process by which a donor cell or otherwise a male cell makes contact with another cell, which is a recipient or female cell. Here DNA is directly transferable. So plasmid carry genetic information necessary for conjugation to occur. So it is through plasmids. So only cells that contains such conjugative plasmid can act as a donor. Those which lacks that, they become a recipient. Okay, as you can see, bacteria A, bacteria B. Here F plasmid, otherwise F factor. Then through it, then only this bacteria is only capable of forming a pile or conjugation tube. So it goes and mates with the F minus and transfers the genetic element. So transfer of genetic factorial from bacteria A by bacteria B by mating or contact is called as a conjugation. So the ability to conjugate is conferred by F plasmid. Okay. So F plasmid or donor cell or male cells or F plus cells, all terms are same. And then when F plasmid absent, it becomes recipient or F minus cells or female cells. So F plasmids, it has got other names also, fertility factor, otherwise sex factor or transfer factor. So a transfer factor that contains genetic information necessary for synthesis of sex pilus for self-transfer without any other identifiable genetic elements such as drug resistance. It will not have any genetic material such as drug resistance. As you can see, this is transferred, the F plus cells becomes F minus cells. So this is an animation. So you can see here. Yeah. So this goes and conjugates. Sex pilus is formed. This is donor nucleus. Okay, this contains no sex pilus. So now conjugation happens. Conjugation tube is formed, and then on complementary, and then a strand is transferred. So a complementary strand for the plasmid is formed. So now this again becomes antibiotic resistant. Okay. So this is So what happens during conjugation? I have shown you. F plus cells becomes F minus cells. So this is simple conjugation. F factor, one of the strand gets transferred and then a complementary strand is formed from the primary strand. So once female recipient cell now becomes that becomes F plus, minus will become male cell. So the characteristic of maleness or F plus in a bacteria is transmissible or infectious. So now what is HFR cell? So when a F factor, otherwise a plasmid which exists in integrated state with the host chromosome, that is called as a HFR cell. So here you can see when F plus cells, when it is integrated with the host chromosome. Okay. So that is called as the HFR cell. Okay. Now here again, this is the thing. So F plus donor bacterium, F plus cell. So now this becomes F plus cell. So this chromosome now becomes integrates with this plasmid. It goes and integrates with the host chromosomes. So this becomes HFR cell. So this is called as HFR conjugation. So F plus cells, it integrates, it becomes HFR cell. That goes and conjugates with the female or recipient or F minus recipient. So here you can see a portion of the HFR cell, DNA or the genetic element. It goes and integrates with the incomplete F minus plasmid, and that becomes recombinant cell. So you can see here. So here a part of the HFR conjugation, a part of the host chromosome is transferred. So this is called as the HFR conjugation. So this is a difference between simple and HFR conjugation. In simple conjugation, F plus cells become F minus cells become F plus cells, and in HFR conjugation, F minus cell remains F minus, but then when it conjugates to HFR, but it gets a genetic element. So here F plus cell and HFR integrated, this is reversible actually. So this F plus turns F minus into F plus upon conjugation. So HFR is in integrated state. Conjugation with an HFR, F minus rarely becomes F plus. It doesn't necessarily become F plus, but it receives a chromosomal DNA from donor. So this is the difference between simple conjugation and HFR conjugation, and this state is irreversible. Integrated state, it can go to again free plasmid state. I can show you. So here, this is free plasmid, and then it goes and integrates, becomes HFR cell, and this is reversible. Like it goes back. This part of the genetic element gets separate, becomes free plasmid, and it becomes only HFR. Okay. So when F factor reverses from integrated free state, it may sometimes carry with it some chromosomal DNA from adjacent site of its attachment. Such a factor is known as F prime factor or F dash factor, F comma factor. So this is F prime factor. So how it comes, you can see here. So when it reverses back from integrated state to F plus, so this goes, when it separates, reverses, it takes the plasmid part, that DNA will take the donor or bacterial chromosomal DNA along with it. This becomes F prime factor. Okay. So F prime, when this F prime goes and mates with the F minus cell, it is called as the sex duction. So when F prime cells conjugates with the recipient, which is F minus, it transfers along with the F factor, the host DNA incorporated with it. So this is called as the sex section. You can see here F prime factor, it goes and conjugates with the F minus cells. So one strand of F prime is transferred to the recipient cells. So here this becomes again both donor and recipient becomes F prime factor. So this is called as the sexduction. It is not conjugation, it is sexduction. So summary, so F plus cells means a donor bacteria can transform F minus into F plus. It can be episomes, which means able to exist in some cells in integrated states. It can transform the chromosomal gene to recruitment with high frequency cells are known as HFR cells. Conversion of F plus cells in HFR cells is reversible. And when this F factor incorporates some chromosomal gene during reversal state, it is called as F prime. So when F prime goes and meets with the F minus, it becomes another F prime, and this process is called as a sexduction. So these are the different types of cells involved in conjugation. So this is F plus cell. F plus cells means this is with the F plasmid or fertility plasmid. F minus cell is without F plasmid. And in this diagram, it is F prime cell means F plasmid containing few chromosomes. So the blue color is the bacterial chromosome. This is pink color is plasmid. So if it takes up the bacterial chromosome, it is F prime cell. And then HFR cells mean when F plasmid integrates with the bacterial chromosome. These are the different types of cells. F plus cells, F minus cells, F prime cells, HFR cells. So medically important factors can be transferred by conjugation, like polysygenic, and then resistance transfer factor. So collision factor also are transferred. The collisions are lethal to other Enterobacteriaceae, likewise pyosins, diphtheriosins. These are all coded by plasmids. So plasmid transmit cold factor will lead to self-transfer of chromosomal segments. And then what is RTF? It is resistance transfer factor. These are the plasmids which help to spread multiple drug resistance. So this was first discovered in Japan, and it was discovered in Shigella, spread of resistance to following antibiotics like sulfonamides, streptomycin, chloramphenicol, and tetracyclines. Okay. So this is RTF factor structure, which I have told you. And then what does resistance transfer factor plasmid contains? It contains two components, a transfer factor or otherwise RT, which helps in the conjugational transfer and another component is called as the resistant determinant, which is marked as small case. The R is marked as small case to each of several drugs. So RTF, which is, or otherwise it is mentioned as RT, resistant transfer factors or resistant factor. So this RTF plus R determinant are totally collectively known as R factor, which is a plasmid. So R factor is a resistant plasmid which contains RTF component and R determinants. So at a time, this R determinants can be resistant to more than 8 drugs or more than 8 drugs. Okay. So what are the genesis of R factor? It is mainly due to the indiscriminate use of antibiotics in veterinary medicine and even in human beings. So here you should know about mutation, types of mutation, methods of gene transfer, and all these gene transfer methods are called as transformation. What is transformation? What is transduction? Types of transduction. Then conjugation, types of conjugation. What is an F factor? What is HFR conjugation? What is F prime factor? What is sexduction? What is cold factor? And what is RTF factor? And then what are the genetic mechanism in bacteria which helps to spread the infectious diseases? So mainly genetic mechanism of drug resistance is very, very important, because antimicrobial resistance is globally it is a threat now. So which of these factors, here is a quiz, which of these factors does not hold good for transferable drug resistance? Here, simultaneous resistance to multiple drugs, increasing the dose makes the drug effective, combination of the drugs is not effective, virulence is not associated with it. So here, transferable drug resistance is, this is the wrong choice, like increasing the dose doesn't make the drug effective in case of transferable drug resistance. So first you should know what are the types of drug resistance. So it is multiple drug resistance and transferable, sorry, not multiple, it is mutational drug resistance and transferable drug resistance. Sorry, you can change it here, like it is not like, it is like, I can, it is not mutational, it is, sorry, it is not multiple, it is mutational drug resistance. So in mutational it is one drug resistance at a time, transferable multiple drugs, and mutational it is low degree and it can be overcome by increasing the dosage, and drug combination can prevent mutational, and virulence is low, so mutants may be defective and resistance does not spread. But in case of transferable drug resistance, it is multiple drug resistance at a time, it is high degree of resistance, and high dose is ineffective, and combination also cannot prevent the drug resistance, and virulence is not decreased, it is not defective, and it spreads to same or different species. Okay. So here what is how the antibiotic resistance, types of antibiotic resistance, it can be a natural, which is innately certain bacteria resistant to certain drugs. So you should know that from your pharmacology. Like typically vancomycin or glycopeptides are resistance to, I mean, gram negatives are resistance to glycopeptides. Likewise, gram negatives are resistant to, likewise, gram positives are resistant to certain drugs. And then it can be acquired. So how it acquires antibiotic resistance? It can be either by mutation or by genetic transfer. Mutation I have told you before, and then genetic transfer, it can be vertical or horizontal. So vertical is again like generation to generation. So progeny, bacteria can spread the drug resistant genes to its progeny, but horizontal gene transfer is by conjugation, transformation and transduction. So this horizontal gene transfer is a potential threat now. So apart from that, several biochemical mechanisms also can confer drug resistance, which you must have read in pharmacology, like decreasing permeability of drugs, attaining alternative pathways, and it can produce enzymes and inactivate the drugs. So this is a pictorial demonstration of vertical drug resistance. Progeny bacteria gets the resistance through the genetic mechanism, and horizontal gene transfer is by transformation, conjugation and transduction. The principle or the mechanism by which how it happens, I have explained earlier. And then mutational drug resistance is stepwise mutation or otherwise one-step mutation. Stepwise mutation is seen in increased use of penicillin, and one-step mutation, for example, is streptomycin usage. And then it may show low resistance or high resistance. So if tuberculosis is treated with single drug as of only streptomycin, some resistant mutants appear and replaces the sensitive bacteria in due course. So there is an occurrence of multiple drug resistance, MDR tuberculosis. Other mechanism, so use of penicillin created resistance in staphylococcus. This is seen due to transduction. Okay, this R factors transfer the things. So how you stop the mechanism? Best option is restricted use of antibiotics. And then what are called as transposable genetic elements? So they are structurally, genetically, it is a discrete sequence of DNA. It is present in the DNA itself. So it move around in cut and paste manner between chromosomal and extrachromosomal. So this transposon can be present in bacterial chromosome and can be present in plasmids as well. And then with this genetic transfer due to transposition, it is possible in nature, and it is not self-replicating. So it depends on plasmid or chromosome for replication. So transposon and R factor R forms may have evolved as a collection of transposons. Okay. So, and each carrying genes that confers resistant to one or several antibiotics. So, this laboratory manipulation has led to genetic engineering. So, now these are the things which you have to remember: genetic mechanism of drug resistance, what is mutational drug resistant, what is transferable drug resistant, what are the difference between mutational and transferable drug resistance, which is a potential threat, then what are transposon, what are insertion sequence. So now, how a DNA is detected? It can be detected by southern blot, northern blot, western blot, eastern blot. So which is the correct choice? So the correct choice will be southern blot. So in southern blot only a DNA can be detected. I can show you southern blotting, DNA is detected, northern blotting, RNA is detected, western blotting, proteins are detected. So then what is eastern blot? So does it exist? Is it just for the sake of answer we have given? Okay, I'll explain what is eastern blot later. So, another quiz here. DNA can be detected by all the following methods except choice A is southern blot. The next one is RFLP, otherwise called as restriction, fragment length polymorphism. Third choice is gel electrophoresis, and fourth one is fluorescent staining. So, here the choice which is wrong is fluorescent staining. This doesn't help in detection of DNA. All the other three helps in the detection of DNA. So now we go to the molecular genetics, which is analysis and manipulation of DNA using biochemical and microbiological techniques. So molecular genetics, otherwise called as, these are the different components. Genetic engineering, we have to know about restriction endonucleases, about DNA probes, about blotting techniques, about polymerase chain reaction, then genetic mapping. So what is genetic engineering? It is isolation of genes coding for any desired protein from a microorganism or a cell of higher life forms, including human beings, and their introduction into a suitable microorganisms in which genes would function, directing the production of specific protein. So these are the four modern application for pharmaceutical production of insulin interferon, and for genetic engineered plants, animal gene alteration for production of gene probes and DNA fingerprinting, and human genome initiative. So which of the following statements about restriction endonucleases is not true? So the enzymes are natural products of bacteria. The enzymes cut DNA at defined sequences. The enzyme cut only single-stranded DNA molecule. The enzymes are named because they restrict the growth of bacteriophage. So which is the not true one among the choice? The fourth choice. The enzymes are named because they restrict the growth of, this is the wrong choice. So all the other choices are correct. And then what are restriction endonucleases? So they are nothing but an enzyme that cuts a double-stranded DNA. So the enzyme makes two incisions, one through each of the sugar phosphate backbones of the double helix without damaging the nitrogenous bases. So they work with cutting up foreign DNA, which is a process that is called as splicing. Okay, so restriction endonucleases made advances in genetic engineering. So you can cut and paste. So next is DNA probes. They are radioactive biotilated probes, otherwise labeled copies united with single-stranded DNA. So this DNA probes contain 20 to 25 nucleotides, and then it helps detection of homology of DNA by hybridization technology, and it helps in diagnosis of infectious diseases, and even a minute quantity of DNA is detected. Next we go to blotting techniques. As I have told you, DNA fragments obtained by restriction enzyme digestion on a separation by gel can be transferred to nitrocellulose or nylon membranes. There are several methods of blotting techniques: southern blotting, northern blotting, western blotting. I told you southern blotting detects DNA, northern blotting detects RNA, western blotting detects proteins. So western blot is actually analytical technique used to detect specific proteins, and then it uses a gel electrophoresis to separate the native or denatured proteins by length of the polypeptide or by a 3D structure. So you must be aware that western blot is used to detect HIV proteins. Okay, you can see this. So after that, these things or proteins are transferred by gel electrophoresis on a blotting paper. These blotting paper is cut into strips, and then protein mixture is separated and then blotted onto nitrocellulose strips, and then you can use enzyme label by ELISA method. So when it is homologous proteins are present, it gives a color change. You can see western blot for detecting HIV proteins. Okay. So now, as I told you, what is Eastern blot? Does it exist? Yes, Eastern blot is a technique which is used to analyze protein post-translational modification. So after the protein is formed, if there are any post-translational modification, that can be detected by Eastern blot. And actually it is a modification of Western blot, and then it used to detect lipids as well. Okay, so often it is used to detect carbohydrate epitopes. So Western blot to confirm HIV infection may be landmark. So it is a specific test for detection of, or confirmatory test for detection of HIV or AIDS. So next we go to PCR, otherwise polymerase reaction. The polymerase chain reaction, which is detected by Carey M. Mullis in 1983. It is a rapid automatic amplification, and Nobel, for which Carey Mullis got Nobel Prize winning in 1993. So how this is now everywhere, you must be learning, seeing about PCR, particularly RT-PCR. So RT-PCR is nothing but real-time PCR. So actually this is a type of PCR in which is actually modification of the original PCR technique. So here there is amplification of the nucleic acid, whether it can be DNA or it can be RNA, anything. So if I have to take class, so it will be like a separate class for PCR only. So I can put forth if the students are interested, I can take a separate class on PCR, particularly in this COVID situation. People have to, students have, there might be many questions which are related to RT-PCR for detecting COVID or otherwise coronavirus. So if it necessary, you can let me know so that I can take a separate class. So PCR contains of several cycles of sequential DNA replication. So first cycle becomes the template for the next cycle. As you can see, uh, it makes available of abundant quantities of DNA or RNA, and finally it can be detected. So here there are annealing, then first it will be like denaturation, then annealing, and then amplification. Each occurs at a different temperatures. Okay. So ultimately, after a set of some 35 cycles or 40 cycles, you will have an amplified nucleic acid from the primers. Primary nucleic acid will be amplified many a times, logarithmic multiplication. And next is genetic mapping. Here genetic sequences are mostly mapped, like for bacteriophages or viruses. So this is the latest technology, or otherwise next generation sequencing, or whole genome sequencing, or actually taken from, or otherwise modified technology. So that nowadays these are the gold standard techniques. Slowly, I think all the other technology will be replaced by next generation sequencing, which is a confirmatory techniques. So in this class you have learnt about bacterial genetics. You have learnt about what are plasmids, how bacterial chromosome differs from a um, um, higher chromosomes, and what are plasmids, and what are the different types of plasmids, what are the uses of plasmids, then what are the types of genetic variations, and what are mutation, and how genes are transferred, like different techniques and different methods like conjugation, transduction, and transformation. And in transformation and transduction, what are the types of transduction? Conjugation, what are the different types of conjugation? And what are F factor, what are F minus factor, what is HFR cell, what is the F prime cells? Okay, all these things are explained. So this is about bacterial genetics. Any questions? Ulike, should I wind up the class? So Pulikesh, I have finished the class. Okay, thank you, thank you.